I'm Andy Kogelnik. I'm the founder and CEO of the Open Medicine Institute, and I'm a really confused individual because I'm a physician. I still see patients. Every now and then I'm a patient. I'm also a computer scientist, bioengineer, so I spend a lot of my time thinking about data, and particularly omic data, and how we can combine it and actually use it in clinical settings. And I'm going to talk a little bit about that today. Really how we can use that with a little bit of what Catherine and Caitlin have already talked about, which is combining that with patient data directly, and how do we really use this explosion in patient engagement and as well as the explosion in the biotechnology world to take all this data in one place and use it to actual practical purposes. So to me, patient and clinical data and combining it with omic data really gives you a new challenge in the clinical setting. It can easily overwhelm a physician, but it can also easily overwhelm a patient. And yet, when I sit in my clinic, I have patients who walk in the door and say, "Doc, here's my genome," and they've got this stack of stuff. They've been sequenced somewhere. They've gotten 23andMe. They've gotten a whole genome sequenced from somewhere. What does it all mean? And you're supposed to do this in that 8-minute visit that you have with the physician, with the patient, right? So we're down to 8 minutes with a physician for the average patient visit, which is down from about 30 minutes a decade or so ago. And so you barely have time to say hello, let alone go through an entire genome. But there are actually a lot of things, and I think actually that problem is only going to increase as more and more people get access to their own genomic data. But on the whole, I think that's a good thing. I think we physicians are overwhelmed by this in many ways when it happens, because the tools are really in their infancy to how to deal with this, let alone how to relate it back to the clinical setting. But I'm going to talk a little bit about some of the programs that we've been working on that relate to this and really hopefully help to drive towards the optimization of research, but also of clinical care, which is really the reason why we're all here, is to have a better patient outcome at the end of the day. So I don't think I need to emphasize this slide to this audience, is just that sequencing and genomic capacity in terms of being able to generate huge amounts of data is really just going through the roof. It's going so fast through the roof that actually the expansion and drop in cost of storage capacity on the IT side, actually those two lines intersect right now. So we need to rev up the way we store things and our capacity for storing things, because right now on the genome front, we're actually generating data at a rate that's soon going to exceed our ability to store it, which is a whole nother set of problems, but one that's probably of strong interest to this community. So we're not just talking about sequencing data here, though, and I just want to remind the audience that we, while DNA data is interesting and extremely useful in some ways, we're talking about other omic data as well, which have similar slopes to this curve. We're talking about proteomic data, we're talking about RNA expression data, and the genome is just the blueprint. And as Randy Scott, who's on our advisory board, once said to me, he said, the Human Genome Project and having the sequence really for the first time let us know what the questions are to ask, and there's a lot of them out there, obviously still to be asked. So I think as you heard from Catherine and from Caitlin, and I'm sure you'll hear it from Ben as well, and we're going to drive this home that there's a power of data in the crowd. And for patients in particular, I think Ben and Jamie Haywood's company, has leveraged the crowd capacity of patients directly. But what we're seeing now is that there's a whole nother movement out there around wearable devices, which Caitlin referenced, right? The various wearables, and omic data. Right? So how can we harness all this data? And I think Catherine's effort with QIAGEN is a huge one to start to drive patients to look at their own information, but also to make it available to a broader group of people and hopefully a broader set of tools that will help us churn through the meaning of all these things. Whoops. I don't know where that slide came. Oh, I see. Looks a little different on my computer. Anyway, so wearables obviously have exploded. We've taken these now actually and put them in the same context with some omic data. So the ability to collect data, we now have actually a wearable. It's not the Apple iWatch, but from another company in Silicon Valley where we live, that monitors 40 biometric parameters continuously at once, right? So you get a data stream that's continually monitoring across patients. And we've actually used this now in a context where we're doing serial gene expression panels. So these are AFI gene chips here, HDA chips, and correlating that with functional data. So how far do they walk? What was their heart rate? What was their heart rate variability? And actually looking at particular problems. This crew has orthostatic blood pressure issues, and looking at the gene-by-gene impact of various influences, treatments or otherwise, on those patients. So it's an incredible amount of data that you can generate just at one instant, let alone longitudinally through the course of a patient's either treatment or experience. So Catherine kind of hinted at this, but I'm going to go at it from a slightly different perspective. But one of the key pieces is integrating that data and making it accessible and shareable. We've kind of gone down the route of the Google mashup. So we really need, and as I'm sitting there in my clinic, seeing a patient, I need a way to look at a sort of a holistic view across this patient. And by holistic here, I mean holistic not in the sense of a style of medicine, but really in a style of data, right? Looking holistically across every possible channel of information that I can across that patient and trying to have that summarized for me. So I'm going to, of course, look at the typical clinical data. Whoops. The typical clinical data that we might see in a physician's office, so blood pressure and heart rate and things like that. But also any imaging that's coming in, pharmacy data, device data, insurance data, omic data, and we've actually added to this a whole bunch of functional data types. So it turns out your financial data actually has bearing on your health. It turns out that activity data, obviously, all kinds of data can let us profile you as an individual and make predictive assumptions about you or current analytics about you and your health. We can identify, actually, in particular on the mental health side even now, changes that correlate with certain disease states. I'm sure all of you have heard about some of the Parkinson's work where people were looking at eye movements and the inflection of voice. And so actually speech recognition patterns or sound wave, actually more specifically recognition patterns, can be used to predict, in part, early onset of things like Parkinson's. But imagine now combining those kinds of technologies with omic technologies where we can monitor at a more closely at a molecular level. So we're trying to use all of these data and sort of mash it up into one dashboard so a clinician or even the patient themselves might be able to start making predictions and or plans around their treatment. So I'm going to give you a few examples now of some of the projects that we've been working on more recently. The first one is a collaborative effort between Alchemy, which is the Adario Lung Cancer Medical Institute, a nonprofit in the lung cancer side, the Dana-Farber Cancer Institute, and USC Cancer Center, and some things went off the edge of the slide here, but we've involved with this, obviously, a whole bunch of community physicians, obviously an incredible number of patients, and then groups like Foundation Medicine were involved as well in this. So this was a study of cancer patients, specifically lung cancer patients, and as the name implies, young lung cancer patients. So these are patients who typically hadn't smoked, and really, from our cultural, social mindset and clinical mindset in some ways, had essentially no risk factors for lung cancer. So why did they get cancer? Well, nobody's ever really looked at this, and nobody's really ever looked at the molecular side of it, in part because it's actually really hard to catch. So there's recruitment issues on the research side, there's a practical issue on an omic measurement side, and there's a participatory issue on the patient side. These patients are frequent enough that it's actually a huge problem in the lung cancer field, but they are seldom enough that most of them sit out in community hospitals and don't always necessarily make it to a big academic medical center to be sequenced or evaluated or otherwise. So we launched a remote consent community protocol, which basically allowed patients to enroll online initially or through a number of different centers, but the majority actually of our enrollment wound up coming online. And we developed an entire accessioning and registration process through our Open MedNet platform. And with the Adair Lung Cancer Foundation, we launched this study, and through basically a viral campaign that was largely patient and physician driven, the dotted line here is a normal clinical trial uptake enrollment pattern. So you have sort of a slow start, and then things kind of after a couple weeks to a couple months get rolling, and then you have a little inflection point where you've got a number of sites up and running, and eventually after 6 to 12 months you reach full enrollment. Well, we went and did this little viral campaign, and it really took off. As you can see, we reached full enrollment within 3 weeks, which was just phenomenal. I mean, none of us had expected this. So this is getting full clinical questionnaires filled out by patients, getting samples, both blood samples as well as tumor blocks, into a specific pathway so that they would all be sequenced the same way and that we could truly look at the genomics of these cancers. This study has now gone international, so we actually have some participation from Europe and now from Asia as well. But it's just really been an education for all of us in terms of how to run a clinical trial and how to do so remotely in the clinical setting, which is in the community setting, which is a lot of what OMI spends its time doing. All right, so we also look at, I'm going to shift gears a couple of times here just because I think there's a lot of really good illustrative examples. I won't go too deep into any of them, but I'm happy to take questions on them afterwards. There's a lot of work actually coming out now around poorly understood diseases. So chronic fatigue has been in the news a lot, which is an area that we've spent some time with. Lyme disease and autism. So these are all sort of related diseases around immunologic function in some way, but not really very well understood. And autism has been in the news a lot in the last 2 years, with autism rates in kids going to 1 in 55 kids, which is just an incredible rate if you think about it. This is a disease that, or a condition that a decade ago was less than 1 in 1,000. So why all of a sudden do we have 1 in 55 kids having these kinds of issues? There hasn't really been, again, an omic, there's been a huge outpouring of patient support for these kinds of things, but we haven't really connected the science and the omics, and part of that comes to funding, which we'll leave to a different discussion. But so we've embarked on a couple of community-based studies, actually almost a dozen now in these various areas. This is just a map of one of those studies, which again we did mostly via the internet, but with direct sites participating, so there are actually clinical evaluations at sites all over the globe where patients are, again, taking and giving longitudinal information, both that we request of them in terms of questionnaires, specific validated scientific instruments, but also patient information similar to probably what Ben is going to talk about in terms of patient-generated data of what they feel is relevant for their condition, and combine that with actual clinical evaluations and obviously longer and more in-depth molecular evaluations. So this is just a sampling. In a very short time, we've tracked a couple thousand people. Actually, this is a subset of it over about 4 years now, and learning incredible amounts of information about how these diseases evolve and potential biomarkers for them. So this is just one example of, longitudinally we can start to separate some of these patients out, and you can see two clear subpopulations here where we've actually distinguished. This is actually a group of chronic fatigue patients, and you can actually clearly draw a line here where there's been a shift from some of these patients on mostly immune genes. This is a gene expression panel turned sideways. So patients are the rows here, and the columns are different genes. And you can, I'm sure most of you are used to looking at heat maps, but clearly there's at least two different patterns here. So we have two different subgroups in a group that was clinically looked at as similar. This bottom panel, just as a reminder to tell me to talk about a little bit more about proteomics. So we're doing profiling across lots of different omics, microbiomics, proteomics, metabolomics, and so forth. This panel actually is done by Applied Proteomics, which is a company out of San Diego that gives us 300,000 data points of 300,000 different protein features for one sample. That's a heck of a lot of data on one time point for one patient. Add that to some of their genomic sequencing data, and we're going to be analyzing this for a while. So if any of you out there have some ideas about data and analytics, we're always open to collaborations. But we actually are working with a lot of groups, including actually Ph.D, who I think is presenting elsewhere at this conference, to sift through this data in different ways and actually finding various outliers and various groups of interest in terms of genes. So we're also looking at larger scale screening efforts, and the two I wanted to highlight here are pregnancy and rare diseases. So we work with a lot of different rare disease foundations. One of them is RGI, Rare Genomics Institute, and in collaboration with them, we're collecting basically N of 1 type studies, but often these turn into N of 12, N of 20 type of collections around what turn out to be very rare diseases. And as one of my colleagues is fond of saying, that all of us have a rare disease. And if you look really and think about personalized medicine, that's really true. It's just a question of how clinically relevant and how far up the clinical scale it goes. Is it something that you're willing to live with? Or is it something that actually impacts you, and that's when it really becomes a disease per se? So the question is, when does that get expressed in your lifetime, and how severely does it get expressed? So with this, again, sort of my mantra here is more data, but it has to be good data, quality data, and so we do spend a lot of time sifting through not only the omic data, but also the patient data to determine what is quality data and what do we actually want to pay attention to. So more data equals better knowledge equals better action, and I'm going to come back to the better action part because I think actually, as Ketan suggested, we're actually already there where we can apply a lot of this information back into the clinical context. Maybe not in one day, but Ketan, we'll get there. We're certainly down into less than 1 week in many cases, which is exciting. So on the rare genomic side, we can take an individual and put them through a pipeline that actually gets an individual patient sequenced, and us in rare genomics, we've found actually several important biomarkers for these rare diseases and actually changed the course of that patient's treatment as a result. Which is really exciting that people can come into a system, and particularly come in from the community, not even coming in necessarily from an academic setting where you can go into a local community hospital or even a local physician's office and start to have this happen. I didn't talk about pregnancy. Let me talk about pregnancy for a second. So together, we've formed a group. It's a collaborative group through DIA. Which is a global drug information association, around medicines and pregnancy. And this is one of the areas where I think the screening idea becomes really critical. So one of the challenges with pregnancy and that women have in pregnancy, besides the pregnancy itself, which is always a huge change and a huge challenge for most people, is that about half the population now has a chronic disease. So imagine what happens when a woman who has a chronic disease and is on a medication for it, say rheumatoid arthritis or diabetes, or pick your disease, there's plenty of them out there, and they become pregnant. They now have to make a decision around that medication. Turns out that most medications actually don't have a whole heck of a lot of data around their usage in pregnancy. So that woman has to make a decision along with her physician, her spouse, and whomever, and oops, I guess that's my time signal here, around whether they're going to stay on that medication or not. And that is incredibly difficult. So what we're doing there is developing a process by which we can capture that information because there's plenty of women who've gone through that before them. We just have not captured that data, and we haven't put any omic data together with that. So I'll come back to that maybe in the panel. Last thing I wanted to touch on was sort of the application and action. So we've actually, together with a few different healthcare systems and insurers, are starting to look at population health and actually applying omics into these populations. In our first year of doing this with a population of 10,000 people, we actually wound up saving that health insurer $2 million in ER costs alone, simply by looking at digital health and genomics and applying them into a clinical context, which to me was just phenomenal. So we're now doing this in a population of 300,000 people and steadily trying to grow that. So if you're interested in that, move to California and we'd be happy to include you in the cohort. But we'll hopefully be doing it other places shortly as well. So lots of people to thank and lots of groups that have helped us along the way and really a lot of collaborative efforts, and happy to take questions afterwards or in the panel. Thanks so much.